DNA replication, proteins associated with DNA replication. Initiator protein, DNA unwinding protein or helicases. Accessory proteins, then untwisting proteins. So unwinding proteins or topoisomerases are DNA strand separating proteins. DNA strands separation creates topological stress in helical DNA. The stress derived by topoisomerases. These enzymes cause reversible breakage and rejoining of DNA strand. There are two types of topoisomerases. Type 1 topoisomerase break just one strand of DNA. Type 2 isomerase introduce simultaneous break in both the strands. Earlier they were named gyrases. The breaks introduced by both types of this enzyme serve as shivels. This allows the two strands of template DNA to rotate freely around each other and prevent overwinding of DNA. These enzymes are essential in the replication of circular DNA so that its replication can proceed without twisting the DNA ahead of the fork. In eukaryotes also, these enzymes are required, otherwise the complete chromosome would rotate during DNA replication. Single-strand DNA binding proteins or SSBP. The separated strands of DNA helix are stabilized and protected from the hydrolytic cleavage of phosphodiester bonds by DNA binding proteins. These are also called single-strand binding proteins or SSBPs. Primase are a specific kind of DNA-dependent RNA polymerase. They function only during DNA replication. Primases are specific kind of DNA-dependent RNA polymerases. They function only during DNA replication. They synthesize a small piece of RNA called RNA primer, which serves as a primer. They synthesize a small piece of RNA called RNA primer, which serves as a primer on which deoxyribonucleotides are added and a new polynucleotide strand complementary to template strand of DNA is synthesized. It means primase can initiate formation of polynucleotide strand, which DNA polymerase cannot. In E. coli, each primase functions only in association with 6 other proteins. These proteins are collectively called primosome. The other proteins of primosome are believed to be associated with the unwinding of specialized DNA and recognizing the different sequence or origin of replication sequence. In eukaryotes, primase is not closely associated with the unwinding proteins. It is tightly bound to DNA polymerase alpha. Next is about replication models. Following models of DNA replication have been identified in prokaryotes and eukaryotes, which are the theta replication of DNA. This model of DNA replication was studied in the circular DNA of bacteria E. coli. Replication begins with the unwinding of DNA double helix at initiation point, also called origin of replication. An initiator protein binds to this point. Using energy from ATP, the initiation protein slightly unwinds the double helix at the initiator point, separating the two strands. This produces two forks called the replication forks or Y junctions. DNA replication proceeds simultaneously on both the strands and in both directions from the initiation portion in a semi-conservative fashion. The replication forks are the ends of replication bubble that grows in size as replication continues. The unwinding and replication move along the circle, leading to the formation of two new circles. Each circle has one maternal or template strand and another newly formed strand. Direction of DNA synthesis, helicase A, then replication fork, theta replication of bacterial DNA in E. coli. In this replication, the circular DNA molecule does not break. It maintains its circular form throughout the replication process. However, in the replication of circular DNA, an immediate theta structure is formed, similar to the shape of Greek letter theta. Just put a line inside a 0, I guess. That's how Greek letter theta is shown. And therefore, DNA replication is a circular chromosome in prokaryotes is called reta replication. It is also seen in circular DNA of plasmids, mitochondria, chloroplasts, and some viruses. Each point of origin and the area of DNA is that subsequently replicated forms one replicate. Next, rolling circle model. Rolling circle model. Rolling circle model of DNA replication occurs in F negative or HFR cells of bacteria, in F minus plasmids, and in some bacteriophages. To start with, the nick or basis break is formed in one of the strands in a basic specific endonuclease with the help of specific endonuclease. This opens up one of the two strands of DNA helix. The open strand form 3' OH and a free 5' PO2 end. The complementary closed strand of circular DNA serves as a template, and DNA polymerase adds nucleotides to the free 3' end in the open strand. There is no need of a primer because the original break produces a primer configuration 3' OH. As nucleotides are added to 3' end of the open strand, the unbroken circular template stands rules and the 5' PO4 end of open strand is displaced as tail. Since the molecular shape created in this replication resembles Greek letter sigma, the rolling circle replication has been called sigma model of DNA replication. Sigma model of DNA replication. On completion, the resulting double helix separates from the tail by enzyme nuclease. The enzyme DNA ligase then joins the ends of replication circular strand, completing circular double helical DNA. Simultaneously, the PO4 tail is replicated in discontinuous fashion, forming a double-stranded molecule. Such a structure is called concatemer. The two ends of replicated tail can also join to form a circle by enzyme DNA ligase, or the linear molecule is packed into phage head. Rolling circle model of sigma model. Sigma model of circular DNA in a bacteriophage. Single strand nick occurs, broken strand separates and is displaced by the synthesis of new strands started in 5' to 3' direction on circle, displacing the 5' tail. And a discontinuous synthesis of now also occurs in the tail. When the tail is complete, it can be removed by a nuclease. The linear DNA can then be rolled into a closed double-stranded circle. Linear DNA. The concatemers are essential intermediate in phage production during bacterial mating. In linear DNA molecule is formed during replication process from the donor cell. It is passed on to the recipient cell to complete conjugation. Next is about D-loop model. This type of replication of DNA is found in circular DNA molecules of chloroplasts or mitochondria. Chloroplasts and mitochondria. In the origin of replication is located at different points on each of the parental template strands. Replication begins on one strand and its replication of origin of replication displacing from the other. The displaced strand forms a displacement loop or D-loop. Displacement loop or D-loop. Replication continues until it passes the origin of separation. Replication on the other strand. Replication is then initiated on second strand in the opposite direction. This results in the formation of two circular D-loop molecules or D molecules in each species. Mitochondrial and chloroplast DNA have multiple D loops. First, origin and D loop. Synthesis of new strands on main strand and D loop formed. Synthesis of new strands of DNA on main strand and D loop. Completion of DNA replication, two daughter DNA molecules. Displacement D loop model of DNA replication in mitochondria and chloroplast. Inhibitors of DNA replication. A variety of substances inhibit DNA replication. These act at various levels during DNA replication. For example, extension DNA inhibitors. Some inhibitors interfere with nucleotide biosynthesis. Some inhibitors interact with the DNA template. Nucleotide analogs also act as inhibitors in DNA replication. Some inhibitors bind to replication proteins. Some drugs and antibiotics interfere with DNA replication. Some inhibitors interfere with nucleotide biosynthesis. Some inhibitors interfere with nucleotide biosynthesis. Continuation of the previous video about inhibitors of DNA replication. Some inhibitors interfere with nucleotide biosynthesis. Some inhibitors bind to replication proteins. Some drugs and inhibitors interfere with DNA replication. Substances like methotrexate and fluorodeoxyuridylate interfere with nucleotide biosynthesis. Methotrexate is used for the treatment of cancers. It inhibits actions of dihydrofolate reductase, causing accumulation of dihydrofolate in the cell. High levels of dihydrofolate inhibit following two reactions. Dihydrofluoride, so are the two reactions. DUMP, that is deoxyuridine monophosphate, plus N5N10 methylene tetrahydrofolate gives DTMP dihydrofolate PRPP G glutamine gives PRA plus L glutamate plus PPA. Therefore, methotrexate causes decrease in the levels of DTTP, DATP, GTP and is used in treating cancer. 5-fluorouracil 5-FU also inhibits the activity of thymidylate synthetase, causing depletion of DTMP and DTTP to the cells and consequently DNA replication. Therefore, 5-FU is also used as a powerful anti-cancer drug. Inside the cells, 5-FU is metabolized to 5-FDUMP, 5-fluorodeoxyUMP. It binds to thymidylate synthetase and N5N10 methylene tetrahydrofolate, forming a tight tertiary complex. This bond cannot be broken because of 5 fluoro groups on the pyrimidine ring and thymidylate synthetase get inactivated. Replication of single-stranded DNA and RNA. Viruses with single-stranded DNA or RNA as genomes also synthesize complementary strands using their single strand as template. The complementary strand in turn is used to synthesize its complementary strand. This second complementary strand is identical with the parent strand. During replication of single-stranded DNA of double-stranded DNA molecule is formed as intermediate of a transient molecule. Double-stranded template, old strand, new strand, old strand. Replication generates two daughter duplexes, each containing one parallel strand and one newly synthesized strand. And replication of double-stranded and single-stranded DNA molecule. Then single-stranded template. Single parent strand is used to synthesize complementary strands. Complementary strand is used to synthesize copy of parent strand. Then in retroviruses, single-stranded DNA synthesizes single-stranded DNA with the help of enzyme reverse transcriptase. This process of synthesis of single-stranded DNA from RNA by complementary base pairing is called reverse transcription. The single-stranded DNA is then converted into a double-stranded DNA. This double DNA, duplex DNA, then gets incorporated into host DNA and becomes an integral part. This is seen in parasitic RNA virus, which produce various diseases in the host.